Hi, I'm Eric Sobie from Mount Sinai in New York, and I'm excited today to talk to you about the computational work that we've been doing to address this question that's posed at the top. Who is at risk of a drug-induced arrhythmia? Now, with this being the CiPA group, I don't need to go through a lot of background about the causes of drug-induced torsade de pointes, but of course, it's primarily caused by block of the hERG current, or the Icahn rapid delayed rectifier current. And so the general conceptual model is that hERG block leads to action potential prolongation, which at the organ level corresponds to QT prolongation, and that increases the risk of torsade. But of course, there's complicating factors that make this a little bit more complex. Drugs block multiple ion channels. Arrhythmias can arise without substantial action potential prolongation. So there's been a lot of work to look for other metrics that can predict drug-induced arrhythmia risk. And what I'm going to be talking about in the remainder of the presentation today is the fact that even dangerous drugs cause arrhythmias in a small percentage of patients. So what can we do to identify those patients who are going to be most at risk of getting a drug-induced arrhythmia? We can think of multiple factors that may predispose individuals to ventricular arrhythmias. So it's not just the drugs, but it's also sex differences, genetic mutations, electrolyte imbalances, heart damage, autonomic changes, etc. These things all combine together so that drugs can cause action potential prolongation to the cellular level, QT prolongation at the organ level, and that can increase the risk of getting drug-induced torsade. But although these factors can be said to promote arrhythmic activity, the actual response is highly variable, especially since arrhythmias are rare occurrences. So what we want to address today is who's susceptible to a ventricular arrhythmia. More specifically, which groups are at the greatest risk, which individuals within a group are at the greatest risk, and what are the physiological mechanisms that can trigger this behavior in some individuals, but not in others. That's what we're going to be talking about in the remainder of the talk today. The strategy that we use to address this is to combine mechanistic mathematical modeling with machine learning. So with the cellular mechanistic action potentials that a lot of us are familiar with, we build populations, we apply triggers to those populations, and some individuals get arrhythmic activity, others don't. Then we use machine learning algorithms based on the simulation data to determine the most important features to predict the risk. But what's really critical here is not just that we do machine learning on the simulation data, but it's the feedback from the machine learning to the mechanistic mathematical models. And that feedback allows us to address questions like, what mechanistic differences explain good classification versus less good classification? And what simulation protocols are going to be able to improve our prediction? So what I'm going to, with this being a pre-recorded talk, I'm going to be going pretty fast through this. The good news is, of course, because it's recorded, you can go back and look at it again. But also that I'm going to be talking about two studies that were recently published. So if you're interested or if there's something you missed, you can always get more information from the publications. First, proarrhythmia caused by potential COVID-19 therapies, published in CPT PSP earlier in 2021. And then much more recently in September, we published a paper where we used machine learning to identify predictors of susceptibility. And that was published in PNAS, as I said, in September. And I should acknowledge the people that really were instrumental in this work. Mira Varshnaya was a PhD student in my laboratory who graduated earlier this year. She's the first author on both of these papers. And special thanks to Shui-Yen Mei, who's a PhD candidate in our program, who's a real machine learning expert. And when we started this, we knew a little bit about machine learning, but Shui-Yen helped us set up the pipeline. And now that with her help, we understand this a lot better than we did back at the beginning. So I don't need to go through a lot of detail about the particular mathematical models we use. We are performing our simulations with the famous O'Hara-Rudy model from 10 years ago. But we are now beginning to think of these models as not just a system of differential equations, but it's a tool to combine a whole bunch of model inputs, not just the mechanisms, current through these ion channels, pumps, and transporters, and ionic homeostasis, but also experimental protocols, such as what rate are you stimulating the cells at, elements of the phenotypes, such as sex differences or structural heart damage, and then, of course, the drugs that block particular cardiac ion channels. All of these factors combine together to produce the action potential, voltage is a function of time, calcium transient, calcium is a function of time, And of course, we care about the action potential because that correlates with the ECG on the organ level. And we care about calcium transient because that's the signal that enables contraction of the cardiac myocyte. And those of you who are familiar with our work know that we don't just simulate the published model with the baseline parameters, we simulate populations of models. So now we have a whole family of action potentials and a whole family of calcium transients. And what's important for the results I'm going to show is that we vary the channel properties to generate these populations, but we vary different channel properties, not just the protein level, increased or decreased ion channel expression and conductance, but we also vary in the later simulations, the kinetics, which can mean faster or slower gating of the channels, or shifts in the voltage dependence of activation or the voltage dependence of inactivation. So it's going to be a whole variety of parameters that we're going to vary to generate these populations. Okay, so now I want to talk about the specific results. I'll start by talking about the study that was published earlier this year, proarrhythmia caused by potential COVID therapies. And as most of you remember, at the beginning of the pandemic, there were a lot of drugs that were proposed to potentially treat COVID-19. Some of them, pharmacologists, clinicians knew were associated with potential arrhythmia risk. And so people were saying, well, these drugs might work to treat COVID-19, but you're going to have to be careful with some of them. And what we noticed by surveying the literature is that the effects of several of these drugs on how they affected cardiac ion channels had already been published. This study here from a group at the FDA profiled 30 different drugs, and it just so happened that 4 of them, azithromycin, chloroquine, lopinavir, and ritonavir, were proposed to potentially treat COVID-19. So we already had data in hand about how they blocked these 7 important cardiac ion channels. We had IC50 values and we had Hill coefficients, all measured under standard conditions in this one paper here. And that allowed us to simulate the effects of these drugs, these potential COVID-19 therapies on cardiac myocytes. And we used phenotypic parameterization to develop virtual populations. So we simulated the effects of these drugs in 4 different groups: healthy male, healthy female, heart failure male, and heart failure female. And these are showing representative action potentials from these populations. The references, sex differences were imposed using data from Colleen Clancy's group here in this first paper, and heart failure was simulated with parameter changes that were published by Beatrice Trenora's group in this paper down here. And in these 4 populations, we simulated either chloroquine plus azithromycin or lopinavir plus ritonavir. We did some pharmacokinetic modeling as well in this study to say what would be the standard concentrations that would be seen in the hearts of these patients, and then what would be the effects of those particular clinically relevant concentrations in these 4 populations. The question is, do we observe any population-specific arrhythmic activity? And the answer is yes. More precisely, action potential prolongation is most pronounced in the heart failure female population. So here we have results from lopinavir plus ritonavir. Here we have chloroquine plus azithromycin. First thing to notice is the difference in the scale. This one only goes up to 15 milliseconds, action potential prolongation, or delta APD. This one goes up to 400 milliseconds. So this is a pharmacokinetic effect. Lopinavir plus ritonavir, as it was proposed for COVID-19, was at low enough doses that this was going to be safe. On the other hand, chloroquine in particular was predicted to accumulate in the heart, and that's why you were getting this dramatic action potential prolongation, and especially dramatic in the heart failure female population. And this asterisk here indicates that 85 cells out of the population of 1,000 that we studied it had arrhythmic activity early after depolarizations or repolarization failure in response to these drugs. So then the question became, what factors promote arrhythmia formation in these individuals? And what we did was we performed multivariable logistic regression. We said, if we know the parameters in all the different cells in this population, and the thing that we're trying to predict is this categorical variable, arrhythmia or no arrhythmia, how are the parameters going to affect which group the cells fall into? And so this is following work that we did to analyze calcium sparks in this paper. And then Stefano Moratti and Eleonora Grandi took that a step further in studying early after depolarizations in this paper here. So with multivariable logistic regression, how can we distinguish between these heart failure female cells that got arrhythmias versus ones that didn't get arrhythmias? And these are the results. These are the parameters that are important down to the ones that are less important in determining which category the cell is going to fall into. And of course, increasing the parameter can either increase your risk of the arrhythmia or decrease your risk of the arrhythmia. So that's why they're in two different colors. Now, the first two parameters are the rapid delayed rectifier current, or the Herd current, and the L-type calcium current. Those results were expected. But what was less expected was that the third most important one is the sodium calcium exchanger, NCX, more important than things like the IK1 or the IKS. And then we dug into our database to say, okay, is this really true? So we picked out two cells that had similar levels of IKR, similar levels of L-type calcium current, different levels of sodium calcium exchange. And sure enough, this one with increased NCX developed arrhythmias, and this one with below normal values of NCX did not develop arrhythmias. So that's indicating that the NCX actually is important in determining that. But the problem with this is that these parameters are generally not accessible, right? It would be great if you knew in a patient or in a cellular experiment what all these levels were, but you don't. So then the question became, can we predict susceptibility on the basis of measurable variables? And that brings me to the second study, where we use machine learning to identify susceptibility predictors, again, on the basis of things that are observable rather than on the parameters, which in general, you don't know. Computational pipeline we used was to build a large population of 10,000 cells, apply triggers to every cell in that population, and some cells got arrhythmias, some cells did not get arrhythmias. Then what we wanted to do is take features from the baseline population, not from the cells after they got arrhythmias, but from the cells before the trigger was applied. Could features from that baseline population predict the labels, predict what category the cell was going to fall into when it when it was exposed to the trigger? We did that evaluation through supervised machine learning. We set up a pipeline where we had 8 different machine learning algorithms over here, and then we evaluate performance with receiver operator characteristic curves that plot the true positive rate as a function of the false positive rate. And the area under this curve indicates the performance of the machine learning algorithm. And that's where we determine the best set of features to predict risk and classify cells in this case. Now, what about the triggers we applied? Well, there were 3 different triggers that we used in this study. And we applied various perturbations, and we tuned the level of these triggers to cause roughly half the cells to form arrhythmic activity. So the first one we looked at was the classic one of blocking the IKR current. We also looked at the effects of increasing the L-type calcium current. And then third, we looked at the effects of injection of a constant inward current at all the times when the cell membrane potential was above -60 millivolts. And sure enough, in that case, in that case, in that case, we got about half of the cells in our population that were susceptible to the trigger, that developed arrhythmias in response to the trigger, and about half that weren't. So that gave us a nice balanced data set, which meant that it simplified the analysis of the machine learning results. So what did we find? Well, first of all, action potential prolongation, corresponding to QT prolongation at the organ level, is only a good predictor for current injection. What we're plotting here are the receiver operator characteristic curves. And what you want is one where the true positive rate goes up without getting too many false positives, and then it goes across here. And then the area under that ROC curve would be equal to 1. That would be an ideal predictor. And so what we see is current injection with an AUC of 0.89. It's quite good just with the action potential duration. For IKR block, for I-calcium increase, it's not very good. Then what we wanted to see is, well, what if we don't just take the action potential duration, but we quantify several different metrics from the steady state action potential at 1 Hertz pacing? So we quantified 8 different potential features from the action potential. And what we see in that case is that prediction improves when we're including more information. It's excellent for current injection. It's better, but still there's room for improvement when we're talking about the response to IKR block or the response to an increase in the L-type calcium current. Then what we did was we added a whole bunch more experimental protocols to see how could we improve the performance of these machine learning algorithms. In the interest of time, I'm not going to go through the results step by step. I'm just going to summarize them here. What can we say about the experiments that are needed to predict susceptibility to each trigger with what we would consider to be excellent performance? For current injection, it's really easy. You only need one metric. You only need action potential triangulation. Then you can get excellent prediction just with that one specific feature. For IKR block, it's a little bit more complicated. As we saw, you don't get good prediction with the features from the steady state action potential at 1 Hz pacing. But if you do a more complex experiment where you block the you block the sodium current and you force the action potential upstrokes to be carried by L-type calcium current, if you take the excitation threshold under those conditions, you can still get really good prediction just with that one metric, but it's a more complex experiment. And then the interesting thing is to predict the response to the I-calcium increase, you need to combine results from different experimental protocols. So you can get good prediction if you take the 8 features from the action potential at steady state 1 Hz pacing, and then you also take the 8 features when you do the same experiment, but you have increased extracellular calcium, hypercalcemia. So if you combine these 2 protocols and all of these features, then you can get good prediction. So this one's really easy. This one's a little bit more complicated. And the L-type calcium current increase is even more complicated. So the question then is, why do different triggers present a different degree of challenge? And that led us to the hypothesis that the degree of difficulty for machine learning depends on how many electrophysiological parameters control susceptibility for each trigger. So then what we wanted to do is to test that hypothesis. And the way that we did that was we used lasso regression to try to quantify the number of ion channel parameters that were necessary to predict susceptibility to each trigger. And sure enough, what lasso regression does is it will progressively eliminate parameters from your multivariable regression model until you only have the ones that are absolutely essential. And sure enough, what we see is that you only need 3 parameters to predict susceptibility to current injection. There's 7 parameters that are involved in terms of getting a good prediction for IKR block, but you need many, many more parameters to accurately predict the response to an increase in L-type calcium current. So easier triggers, such as the current injection, depend on fewer channel properties to determine arrhythmia risk, while the more difficult triggers, such as the L-type calcium current increase, are dependent on characteristics of multiple ion channels. And that's why more experimental features from more experimental protocols are needed to predict susceptibility to the I-calcium increase compared to current injection. Okay, to conclude, we can identify electrophysiological signatures to predict risk, and this is what we're really excited about. We don't need to know exactly what the parameters are. We can extract information from, at the cellular level, the action potential waveforms, and hopefully this means at the clinical level, we can extract features from the ECG waveforms that can predict how that cell or how that individual is going to respond to some future arrhythmia trigger. In terms of what we're going to do in the laboratory, the simulations can prioritize experiments to predict susceptibility. So some experimental protocols are really informative and really useful. Others are less informative and therefore less useful. And then finally, the thing I want to leave you with as a final conclusion is that the feedback from the machine learning is what provides the mechanistic insight. So I'll go back to our schematic here. It's not just a matter of combining the simulations with the machine learning. It's really the feedback from the machine learning back to the simulations that is how we get the mechanistic insight for performing these kinds of studies. So now I'm going to finish. I want to thank you for your attention, and I'm excited to participate in the rest of the meeting and have conversations with people about all the unanswered questions that we have in terms of predicting arrhythmia risk. Thank you very much.